Hi everyone. In this video, we're going to discuss a series of videos. We're going to discuss the pentose phosphate pathway. So this is the overview of the next videos. This one, we're just going to cover some basic properties of the pentose phosphate pathway and then go into detail about its regulation. All right, so where does it fit in the general scheme of things? This is our organizing slide again of all the reactions we're going to be talking about, or we have been talking about in this course. So this is what we're going to be dealing with today. How we get reducing power in cells that do not do photosynthesis, for example. Okay, we saw how plants can make NADPH from the photo part of photosynthesis. By the way, they also do the pentose phosphate pathway. Okay, but let's say in other cells like our own cells, how do we get our own NADPH? So this is where we are. It goes by different names. These days, probably the pentose phosphate pathway is the most common one, but you may see it as the hexose monophosphate shunt, pentose cycle, or the phosphogluconate pathway. They're the same thing. So what happens in this case is we have a 6-carbon unit, our favorite one, glucose, and from that, we're actually going to lose 1 carbon, and we're going to get a 5-carbon unit. That's one product, but the other output, of course, is NADPH. This is the reducing power that animal cells, for example, use to or need in order to do their biosynthesis. What happens to the pentoses? It depends. They can go back into glycolysis, and we'll see how that happens. Or they can also serve directly as precursors for amino acids and especially for nucleotides. One major point I want to make throughout this series of videos about the pentose phosphate pathway is how versatile it is. The cell can really, and we're going to talk about this in the regulation later, but the cell, all these outputs are extremely useful to the cell, and the pathway can adjust so that it produces only NADPH or just pentoses, and you know, it's really extremely versatile. It's a liability in cancer cells because cancer cells love this pathway too, because it provides key precursors for amino acids and for biosynthesis, including amino acids and nucleotides, of course, where they need to make their new genome. So very important pathway. It can be broken down in 2 parts. The first part is the oxidative part, the first reactions of the pathway, where really we're going to see some dehydrogenases, and we're going to get from glucose to pentose and generation of NADPH. And the reaction can be summarized in this way. We have glucose 6-phosphate and NADP plus, so oxidized NADP. And then we're going to get CO2 because we lose 1 carbon from glucose, some pentose 5-phosphate, and we're going to get 2 NADPHs. So this is very important, all right? That's the reducing power. What happens now to the pentoses has to do with the reductive branch. We don't have any redox reactions there in the reductive branch. The pentoses can go back and forth with glycolysis, and these reactions are actually extremely similar to what we have seen in the Calvin cycle. So we're going to play again the sugar interconversion game in the reductive branch of the pentose phosphate pathway, and where we're going to get 3 pentoses and convert them essentially to glycolytic intermediates. So 3 times 5, that's 15. That's the same number of carbons that are contained in 2 6-carbon sugars, like 2 fructoses. 2 times 6, 12, plus 1 triose, 1 3-carbon sugar, glyceraldehyde triphosphate that we have seen many times already. So this is the sugar interconversion game that we're going to play again, and we have already played to some extent in the Calvin cycle. Some history. It has a very long history, actually, since the beginning of the 20th century. And just to remind you, dehydrogenation here is oxidation independent of oxygen. So unlike the oxidations that we saw, let's say, during respiration, this is different. And it has a long history because it can actually be monitored with with color changes of some dyes. So it was easier to study, and a lot was known already, by the 30s, 1930s. So in 1930, in fact, Otto Warburg, who we have already seen, to remind you, Otto Warburg was the one that speculated that, or discovered rather, not speculated, that a lot of cancer cells, most cancers, about 2/3 we know now, prefer to do fermentation, glycolysis and fermentation, as opposed to oxidative phosphorylation. And we know now that this, so in some ways they behave like yeast, because by doing glycolysis and fermentation, they can generate a lot of biosynthetic intermediates. So anyway, so Otto Warburg had also done work in yeast with an old yellow enzyme. And what he and others had found was that NADPH and the oxidized yellow enzyme, which happens to have a color, if you have this, oxidized yellow enzyme and you add it to NADPH, then you get NADP and reduced yellow enzyme, which was not colored. So, it's easy to monitor these reactions because all you have to look for is a change in color. And now we know that in the very first pentose phosphate pathway reaction, glucose 6-phosphate and NADP, they react, and we're going to get gluconolactone 6-phosphate. That's an oxidized form of glucose. And so it's a redox reaction, and we're going to have NADP now being converted to NADPH. So glucose will be oxidized, and NADP will be reduced, and we're going to get NADPH. So it turns out that the yellow enzyme that these people were working on could take NADPH back to NADP+. All right, so that concludes this introductory video. I'm going to continue in the next one with oxidative reactions of the pentose phosphate pathway.